Nice to see everyone this evening. I'm going to, as Nina said, I'm going to show you how to do queries on the website and interpret the results in a little bit more detail than what Alex has already shown you. And I'm going to use some of the examples that you guys sent in. And my next button isn't working. So this is the home page, as you've seen, and this is where you can do most of the queries. These 6 search panes let you access the data in the key ways that most people are interested in looking at the IEDB data. So I'm going to walk you through what you can do in each of these 6 panes. So the very first one is about the epitope itself, and this is what people most commonly search on. They have a peptide sequence and they want to see more information about that peptide. How often was it studied? And when you do that, you can just enter the linear peptide sequence in this box. But I want to point out this extra feature that you can search for similar peptides on either substrings if you enter a long sequence, or different blast match identity levels. And I really want to encourage you to use these different blast match levels because different authors may do their experiments on a slightly different peptide than what you're interested in. It may be longer, it may be a little bit shorter, it may have 1 or 2 residues different from yours. So I always encourage a broader search to start with to see how much data there is. And if there's a ton of data, then you can narrow it. So the next search pane is about the source of the epitope. So if it's a natural peptide or a non-peptidic structure and it's derived from an organism and a larger structure, you can find those here. And so a lot of people will come and they just see all the epitopes from a particular organism. So if you start typing in these boxes, they auto-complete and show you all of the hits using the NCBI taxonomy synonyms that are similar to what you type. And the next box is about the antigen. So if it's a protein, you would start typing the protein name and all the proteins that match the term that you typed will show up. And then the next search box is the host. So this is whose immune response is being studied, whose TCRs, whose antibodies, or what MHC molecules. So human, mouse, and non-human primate are pulled out as radio buttons because they're very commonly searched on, but you can access all of the data for any particular host you're interested in. You can browse the tree to see what sorts of hosts we have data for, and it's any vertebrate for which the data is published, as Alex said, and you can use a common name. So if you start typing dog, that will work. It doesn't have to use the scientific name because we have all the synonyms from NCBI. And then the next search pane is the assays. So the IEDB is divided up into T-cell, B-cell, and MHC ligand assays. And if you leave them all checked, you'll get all the assays for whatever your other search criteria are. But if you're interested in just a specific type of assay, you can uncheck the boxes that aren't interesting to you. And you can also use the Finder to see all of the detailed types of experiments we have and maybe limit it to a particular class of experiments. And I want to also point out the outcome box. So this was the data positive or negative. We capture both positive and negative contexts in the papers because the negative data can be very interesting, but most people want the positive data, so that's checked by default. If you want negative data only or you want to include negative data, check or uncheck these boxes as needed. The next search pane is the MHC restriction. So for T-cell assays, if the authors tell us the restriction, we will capture it. And for MHC ligand assays, there's always an MHC molecule. And the high-level searches on class 1, class 2, and non-classical can be done by radio buttons. But if you have a specific MHC, you can start typing it and it will autocomplete, or you can browse the tree to see what species we have data for. And this changes all the time. If someone publishes new experiments on a species that we didn't previously have, it'll show up in the tree as that data gets published. And the next search pane is the disease. So this is the disease of the host at the time of the experiment or prior to the experiment that was relative and important to their immune response. So if someone had a past dengue infection and the epitopes you're studying are from dengue, then that would be captured, even if they had resolved. And the key radio buttons show you the main types of diseases we have in the database: infectious disease, allergy, autoimmune diseases, but we have many others, and you can browse the tree and find them, and you can search on higher levels, like all drug allergies or just all contact allergic dermatitis, regardless of the cause, or you can navigate through the tree and find the specific diseases if you're interested in those. Also point out we have animal models of disease too. So I'm getting to the queries that were submitted. So the first query I was going to look at is how to see the differences between B and T cell epitopes and responses, and if you want to see even further between CD4 and CD8. responses. So to start off with, first thing we're going to do is look for the source of the epitopes, the SARS-CoV-2. So I'll use the source search pane to enter SARS-CoV-2. And when you start typing, the autocomplete fills out. And when you select something from the autocomplete, you get this little one that pops up. And that means I selected one item in that box, but these are multi-select fields, so I can add to my query. I can add more. And you can add as many search parameters as you want. You can pick something from every box, or you can pick nothing. So once you've completed building your query, you then have to hit the search button at the bottom of the page. And once you've picked everything you're going to pick. And I'm going to start off with a really broad query because I always recommend doing that. So all I've entered was SARS-CoV-2, and then I hit the search. And this is what the results page looks like when you do that. And now I want to talk to you a little bit about the results page. At the top, you'll always see the filters you applied. So this was what my query was, and you can X these out and then search again. And if you X something out, the search button will turn green again to let you know that you're pending a search. And then over on this left-hand side, there's additional filters that you can add from what was on the homepage. We add a little bit more detail here because these are things that people are interested in, but not as commonly as the things that are on the homepage. And our most recent new feature is filter options. So it used to be just one set of potential search criteria, but now we've added very specific ones to the different assay types. So because my query was broad, I landed on what's known as the default page. So it's just a few extra fields that are added to the search panes, but we also have T-cell, B-cell, and MHC that give you specific fields relevant to those assay types. So looking at the default filter options, these are things that we get that were not on the homepage. So if you're interested in these types of searches, go to the results page, and then these additional search parameters show up. So in the epitope search box, it's the same as what was on the homepage, but now you can also search for 3D data for that epitope. So antibody or TCR binding in a 3D structure, or MHC binding in a 3D structure. We have all of the data that's in PDB. And then if you're interested in modifications, post-translational modifications to the peptide sequence that you're studying, you can use this drop down and select those. And in the epitope source box, there's an additional search feature that is to include related structures. So these are analogs, mimotopes, and neoepitopes. So they may not fit your main search criteria. So they may not be from SARS-CoV-2 per se, but they may be related because they're an analog of SARS-CoV-2 epitope. So they're one or two amino acids different, so they're no longer natural. They're not derived from SARS, but you might want to see that data. So you use these checkboxes to include them in your results. And then we add a whole new search pane called the receptor, and this lets you search TCR and BCR sequences. So we have sequence data whenever it appears in the paper with epitope specific data, and we let you search by just retrieve all of it, whether or not you know both alpha and beta chain or heavy and light chain types. And then you can also search if you have sequenced the TCR antibody and you have a CDR3, you have a full length sequence or nucleotide sequence, you can enter that and search our data there and find out what epitopes it's known to bind. And then the last new search pane to show up is the reference one. So this lets you search across the references or the submissions that data came from. It's a particular author you're interested in. If you have a title, if you want to narrow your results to a particular age range, so you can do just new papers. And then if you have a PubMed ID, you can search for that here too. And then if you are on the T-cell filter options, you get some T-cell specific search parameters. So when you're searching in the receptor box, you're always searching across TCRs. And here you can see that the chain types are now alpha and beta. If you're searching in the assays, you're now only searching across T-cell assays. So some of the ones that are more commonly studied show up as radio buttons to help you more quickly narrow your results. And then we also add a search feature to look for only data that is direct ex vivo. So that means there was no in vitro restimulation before the experiment was performed. And then in the image restriction search box, we also add the ability to search for resolution and evidence. So this is how well defined the restriction is, because some authors will only tell us the class level restriction. They use CD4 cells, so they assume it's class 2 restricted. So that sort of information is captured, but also if there's very precise restriction, we will have it. And some of our users said that they wanted to be able to narrow their results to only very clearly MHC restricted data. And then also the evidence is how we came to know that, how the authors made that conclusion. And there's a variety of ways of doing it. So some people don't care, and if you don't care, you don't need to include these in your search. But if you want only data where MHC binding was used to define the restriction, you can then use the evidence to narrow those results. And then in the B cell filter options page, there's things specific to just antibodies data. So when you're searching the receptors, you're only searching across antibody sequences. So you can see that the chain types change to just be heavy and light. And then in the assay search box, it's only searching at antibody assays. So you see some of the ones more commonly queried on, pulled out as radio buttons. And you also get the ability to narrow to isotype. So if you only want IgE or IgA experiments, you can do that here using this dropdown. And this is the last filter option, the MHC one. We add the ability to search specifically MHC assays, and you see the two main assay types, the binding assays and ligand elution assays. And then in the restriction box, we also have resolution and evidence, just like with the T-cell assays. So then now back out looking at the results page. So when you first land on the results page, what you get is epitope tab, and then you can see all the other tabs. And Alex went through these a little bit too. And each tab has headers that are sortable. These little drop-down arrows let you sort by that column. And in most of these tabs, you also get a details or ID column. And that details and the ID give you more information about that row. So each of these epitopes has a lot of data. It's from 17 references tested in 62 assays. So when you click on that detail, you get a summary for that row. So this epitope, you can see all of the experiments from which it came, and if it has the structures, and how frequently it was tested, and how frequently it was positive. So this can be very interesting if an epitope has a lot of different MHC restrictions, or if it has T and B and MHC assays, they'll all show up here. And I wanted to point out that whether or not an epitope is positive or negative in an assay is very context dependent. So you need to look into the details of those experiments. So if they were performed in mice versus humans, or two different mouse strains, or if it was diabetics versus healthy controls, you're going to get different results. So that's why all of the search parameters can be so complicated, because you may want to narrow your results to only look at a specific data type. So then also in each of these tabs, you'll see these little funnels with the plus sign, and that lets you narrow your results right there on the fly. You can click on that and it will only give you results for that particular epitope. In this case, this was how you can get just results for the spike glycoprotein and not the other proteins for that. And the next, each of the pages has an export option. And when you export the data right now, you can export to 2 formats, with and without IRIs. And the IRIs are those ontology identifiers for the terms. But we're working on currently creating new export formats and features and adding more help to the exports. And someone will talk about that in a little bit more detail later. But that is because one of those, something we got a lot of feedback for in the past, that people were interested in that. And then the next step over from epitopes is antigens, and it has the header. And so this will show you, so since my query was on one particular organism, the proteins you see here represent all of the proteins in the reference proteome for that organism for which we have data. So there may be other proteins for a species, but if no one's published epitopes from it, it won't show up. But these are all the proteins from this organism that we have epitopes for, and you can see how many epitopes. And it's quite a large number considering this data is very recent. So there's a lot of epitopes from SARS-CoV-2 being published and put into the IEDB as quickly as possible. So the next tab over the assays, and this, like Alex said, this is the meat of the database. This is where you get all the details and that context-dependent information about when an epitope was positive and when an epitope was negative, and how it was tested. So you can see the host that it was tested in, immunization. And this is a summary of many, many epitope fields that we capture in these assays. And so you can see there's T-cell, B-cell, and MHC subtabs. And there's the header rows with the sortable. And there's the same funnels that you saw previously on the other tabs to filter to things. And then again, the IDs. And each assay has many fields. And so when you click on the ID, you'll get the full details for that row for that assay. In this particular one, I clicked on it so I can show you some of the details. And each assay always starts off with information about the reference. That's the first section you'll see. And it has where the data came from. And then the next section that you'll see as you scroll down the page is about the epitope itself. So each experiment belongs to an epitope-specific receptors. So this is the epitope specific for that assay. And then the next section is the immunization details. So this experiment was performed in a human who had recovered from COVID, and there was an in vitro restimulation step where PBMCs were incubated with the whole spike of glycoprotein. So the next section is about the assay itself. This was a tritiated thymidine experiment. It was done with CD4 cells. The authors named their clone. We will enter those names they provide them, and if they give the sequence, we will also add that. And over here on the side, you see these are the IRIs. These are the ontology link outs for each of these terms where you can get definitions and more information if you're interested in that sort of thing. And the very last section is about the restriction and the assay antigen. So in this case, the restriction is class 2 because they use CD4 T cells. So that was how the authors concluded it. And then I want to point out that the antigen is very often not the epitope. In this case, that epitope-specific T cell clone proliferated in response to the whole protein from a specific strain of SARS. Back out to the results page. The next tab over from assays is the receptor tab. And just like the other tabs, it has a header that's sortable, and it has IDs that give you more details. So when you click on those IDs, you get summaries for that receptor. So for this particular CDR3, it was published in many different papers. And in some cases, the author showed it being paired with a specific alpha chain, and sometimes they didn't sequence the alpha chain or provide that information. But the same CDR3 was shown to bind several different epitopes, frequently an influenza epitope, but also a yellow fever virus epitope, and also a SARS epitope. And then the last tab over, references, where the data came from, and you can export this and have a list of the references that are relevant to whatever your query was, if you just want to list a couple of IDs. But it has the same features as the other tabs, the sortable headers, the narrow feature, the detail features, and a link out to the paper. So back to the query I was trying to do. I was looking for the differences between B and T cell responses. So I want to show you my first query that I had done was just all data. So I have T cell, B cell, and MHC. And now we want to narrow it to just T or B. So you do that using the assay search box. And you can do this on the home page or the results page. By checking or unchecking these boxes, you get just the T cell epitopes or just the B cell epitopes. So I went ahead and did that. I unchecked B and MHC and hit search. So now I have just T cell assays. You'll see there's nothing under the two tabs. So this 1,907 epitopes, these are T cell epitopes. And then when you repeat the query, looking for just B cell assays, you get just B cell epitopes. And then you can export these two sets of epitopes and can compare them. But it is entirely possible for an author or different authors to test the exact same peptide in both the B cell and a T cell context. So you'll have to compare them and see if any of the epitopes overlap between the two sets. And then to take it further, to look between CD4 and CD8 epitopes, you do that in the MHC restriction search box. And again, you can do this on the home page or the results page, and you can click between, by checking class 1, you look for the CD8 response, which I did here. And then by repeating the same query again, but this time checking class 2, now I've got the CD4 response. And again, export these two sets of epitopes and can compare them. And it is possible that the same epitope may give you both a CD4 and a CD8 response. It's completely up to what the authors test and how the experiments turned out. And then as Alex showed you, and Alba will show you again after I talk really in more detail, this Immunome browser feature is a great way to visualize the data in the IEDB. And when you're on the antigens tab, what you look for is this little graph icon, and it's next to each protein because it plots all of the epitopes for that protein along the length of the protein. So it's a great visual aid. Alba will show you how to do this in detail, but I just wanted to point out that you can do your query looking at B cell data and do your query looking at T cell data and plot them on the Immunome browser. Then you can compare what the B cell response looks like versus what the T cell response looks like. And the blue lines are positive data points and the red is negative data points. And this is so totally dependent on what your query was. So I could further narrow this to just be human response or just a human who actually had COVID. Their responses are going to look different, but also it's dependent on what authors do. So certain regions of a protein may have no data points because no one studied them, or there may be a lot of data points because many people found it interesting and kept repeating the experiments. And this is another graph. This is B and T cell responses for hemagglutinin. And in this case, it was humans who had influenza. And you can see how the graphs are quite different. And you can do any queries you want, maybe like bulb C mouse versus a C57 black mouse, or people who have diabetes versus healthy people, and you'll see the differences very visually between the different conditions of your queries. And then back to user queries, I have another query that someone submitted. They wanted to see spike-specific T-cell epitopes that were induced by infection. And so on the homepage, you can do this query picking just T-cell data, because they had asked for T-cell data, and epitopes from SARS-CoV-2. And this time it's really important to say that the donors had an infection. So I start typing for disease, COVID, and I pick the COVID-19 disease so that you know that the hosts had the disease. And then when I land on the results page, I want to add that it's from the spike protein specifically, and I did that by hitting the funnel next to spike glycoprotein, and that will narrow all my results from all the proteins from SARS to just the epitopes from spike. When I do that, you see it appear up at the top, and now I have these 464 epitopes. And you can take the assays with the epitopes and look at them in more detail. And then this, I think, is my last user query. It is, how do we assess the protective capability of epitopes in the database? And so to do that, the first thing we have to decide is what does protective mean? So epitopes that were tested in a protection assay and were positive in that assay. So we look into the assay finder, and this is the tree view of what types of assays we have. And both B and T have a biological activity branch. And in the biological activity branch here, I opened up the one for B-cell assays. You'll see there's all sorts of different assays. This is totally dependent on what people publish and what types of experiments they perform. And I really wanted to point out that for in vivo assays, we do also have assays that induce disease. We have disease exacerbation. So the IEDB has not just infectious disease, but allergy and autoimmunity and transplantation. So you can imagine that some epitopes, when administered, cause an autoimmune disease or an allergy or a transplantation failure. So you have to look through the assays and decide which ones to you are reflective of protection. So all of these challenge assays generally are accepted as evidence for protection, but for some epitopes, there may not be that much data, and you may want to also include some of these in vitro assays that some people feel are good correlates for protection. So if there's not a lot of data already published using challenge assays, you might want to include some of these others, like neutralization or opsonization. So I did a query for SARS-CoV-2 epitopes that either decreased disease or were positive in a challenge disease. And I got 48 B-cell epitopes, and you can see these are mostly discontinuous epitopes. And you can take the assays and export them and look into the details of exactly how those experiments were performed. So Nina, do you want me to go on, or should I pause now?
We are running about 7 minutes ahead of schedule, Randy.
Okay,
so if you'd like to go over a few of your export slides, that would be great.
Say that again. We're over, right?
Under 7 minutes.
Under, okay.
Yes, if you'd like to go for a couple of your additional
100% misunderstood you. Okay, so I have some additional slides, and they were created based on answering other questions people had asked, but I wasn't sure if we'd have time to go into them. So there are things people had asked about. Many of the people that come to the IEDB website want to export the data and then do all sorts of other analysis to look at the details and to do manipulations of the data. So when you export them, and this is an Excel version of that CSV, I want to point out that currently there's two rows. There's a header row that tells you the section, and then the next row is about the field within that section. So these are all fields that relate to the epitope. And this is the epitopes, modified residues, the epitope description, the epitope object type. And then another question that we have frequently received is about the source of the epitope and the positions within the protein. So that for the epitopes, we have both the isoform that 100% blast matches to, and sometimes authors will specify a particular GenBank identifier or PDB identifier. So we do assign every epitope to that isoform that it's specific to, but also we group them all using the reference proteome. So there's two proteins. So these position fields are the position of the epitope within its source. And then for non-peptidic epitopes, we have link outs to KEBI, the KEBI ontology. And then as you move along the Excel sheet, moving out, the next section over from what I just showed is about the antigen, that source. And so here you see the two different proteins. The first one being the exact isoform the epitope 100% blast matches to, and then the next set of fields, these are the reference proteome. And so you can use the reference proteome proteins, the parent names, to group epitopes. So they'll all have the exact same name or the exact same identifier in the reference proteome, even though the isoforms have highly variable names. Like every gen bank for the same protein may have a slightly different name. So you can use these to sort and group them. And the next section over, this is very similar to what I just showed. This is the exact organism name that the author stated their epitope was derived from. And it could be on a high level, like species level, human or hepatitis C, or it could be a very precise strain of a virus. So if the authors claim that the epitope is from a strain, we will always honor that and capture it. But for grouping purposes, all of the epitopes in the IEDB are organized also by species level, which we call the parent organism. So this allows you to group all influenza A epitopes all together and then look at the precise species that the authors or strain that the authors stated. So they're both valuable information. This gives you some level of 100% matching an author's intention, and then the parent gives you a way to group the data. Or if you're not interested, you don't really care about the specific strains, you can consider just the parent organism. This is export source of data. So many people, when they're looking at the data, they want to know where it came from. And so I want to point out in the exports, we have links back to the assay, links to the reference in the IEDB, and the PubMed ID is in the assay export. Then the assay types is another question that we frequently get. People want to look at how was the data generated. So in the export, the single assay type is broken down into 3 columns so that you can sort on the different columns. So for example, if you're only interested in IL-2, you can sort by 1 column and get all IL-2 assays, whether or not it was performed using an ELISA or an ELISpot. Or if you're interested in quantitative data, you can sort by the units and get all of the data that gave you an nanomolar value. And then the IRI, this is an expert with IRIs. You can see that this is an ontology identifier that you can link out to. And then we had also a question about the positions in discontinuous epitopes. So the epitope position that's shown is the position of the epitope in the source that was chosen. And I think that's it for extra slides. So Nina, do you want to start the Q&A? Do you want me to stop sharing?
Absolutely. Thank you so much, Randi. We are smack bang almost on time. So I'd like to thank Randi for the presentation, and I'd like to invite Alex back to kick us off with our first Q&A session of the agenda. So Alex, I believe you've already picked out a question to kick us off with.
Yes, so there was a question from Tanya. The question was, you mentioned important residues on discontinuous epitopes are added to the database. Could you comment on the guidelines for defining such important residues? Are those the ones that decrease affinity of value of X%, or only the ones whose absence completely hinder the binding? So good point. And the answer is neither. In the sense that we tend to curate relying also on what the author themselves define as a significant decrease or important decrease. And so, of course, if something is a mutation completely abolishes binding of an antibody, yes, that's clearly an important residue. In other instances, there may be not a complete loss, but the authors will point out that that is a significant decrease, and that is also curated. In general, in a discontinuous epitope setting, also it's important to distinguish mutations that may impact reactivity, but are not really part of an epitope. So for example, if you have a proline or a cysteine residue, which is absolutely crucial for the protein to maintain its folding, mutations there will just basically completely obliterate the folding of that protein. And typically what happens in those situations, that is noted because that mutation will abolish reactivity to any antibody reacted to that protein. And so that in that case, we don't curate that as an epitope.
Excellent, thank you, Alex. Randi, would you like to take the next question?
Sure. So the question is, do you curate the antibody epitope data obtained directly from commercial antibody development companies? Not if it's published, we curate it, or if it's submitted, we curate it. So commercial antibody development companies do not typically make submissions to the IEDB. I'm not aware of there ever being one, but if the data gets published, so very often the commercial antibody company will make the antibody, but then someone else, a different author, will publish. So we always put the antibody name, that is a field in the IEDB, and you can search for it. I didn't show that, but it's in the receptor search box. You can search for TCR name or an antibody name. So if the authors state that, I mean, they have to, it's required in the methods, materials and methods. If they put the commercial name in, we put that in the IEDB, so you can search for it.
So I go back to another question from Tanya, which keeps asking very interesting questions. And Tanya asks, are all positive or negative assays treated equally? Or do more assays score more than others? Which is one question. And then in the same question, she also asked, what do you do about sample size? A negative epitope that was tested once is treated as the same, but was extensively studied, say 0 was positive in 0 out of 35. So regarding the first questions, are all positive and negative assays treated equally? Yes, they are. On the other hand, as I was showing a little bit in my introduction, the user has the capacity of selecting in their query particular assay type. So that one user may say, well, you know, I really don't think that an ELISA against a linear peptide, it's as relevant as a neutralizing antibody assay. And so then you could say, okay, I just want to query for neutralizing antibody epitopes. And that, so you can do that, but there is no mechanism to weigh some particular assay better than other. And that is in a way at the essence of the philosophical stance of the IEDB, but we did not want to make judgment call and say, you know, this assay is better than other, or this this group is better than other, or this pretty much the same way when you search PubMed for, you know, tyrosine phosphorylase number 5, you don't want to get only the results of the papers that the PubMed staff thought they were pretty good. You just want to get everything out there, and then you can decide which particular record is useful to you. I mean, so we comprehensively curate all the data that is out there, and then empower the user to query and specify the parameters more readily. Now, the second question is something that was tested once count as the same as something that was tested 35 times? Absolutely not. And that's obviously a well-engraved scientific idea. You know, it's something that has been reproduced in multiple studies or in multiple assays. It's more true than something that was seen only once. And in fact, the immunobrowser has a specific feature that takes that into account. While showing the response frequency, so let's say that you have something that was tested and was found positive 1 time out of 2 times that it was tested. You would intuitively trust that 50%, 1 out of 2, less than if something was tested 150 times and was found to be positive 75,150. The immunobrowser calculates an upper and lower bound of the based on binomial probability of the frequency of responses, exactly to give an estimate of how reliable is that on statistical grounds, that estimate of frequency. Randy, your turn.
Okay, so Alex, there's only 2 more questions, and one of them I agreed to answer before, but the second one came after.
Well, then you're stuck.
It's addressed to me, so I was going to answer the one addressed to me, and then you'll
okay.
So the question was, do the 90 to 70% blasts available at the IEDB consider gaps, or are they entirely gapless? So I'm not very good at answering questions about the algorithm, but the blast matching does consider that not each amino acid will match as long as the percent identity comes out to be 70, 80, or 90%. The gaps will, if there's a gap difference, you will get those hits. I don't know if that made sense, but it wouldn't work, say, for discontinuous epitopes. It only works for linear epitopes.
Thank you,
Okay.
Randy. It looks like Bjorn would like to add to that.
Yeah, I was hoping he might.
Yeah, so just for the question of gapless alignment scores. So the current, the alignment that we are using is highly penalizing gaps. So it can take gaps into account, but I wouldn't use it for that. So essentially you can assume it a gapless alignment for all practical purposes. That's it.
So then I'll jump in and try to answer the last question. Is there are more epitopes in the search? It is possible to converge and delete all the duplicates to show the possible epitope interaction. I'm not sure I totally, completely get the question. The duplicates are not deleted, but are condensed in the immunobrowser. If there are multiple, the same epitopes was tested in multiple assays and multiple times, then all that data is condensed. And that is also done when you have a research and you click on a particular epitope under the epitope tab. It will show all the different assays and all the different instances in which that epitope was tested. So I'm not sure whether I answered the question. If I didn't, please put something in the Q&A to explain more or ask more detail.
Excellent. Thank you so much, Alex, Randy. Feel free to clear out the Q&A there for you both, that looks like.
Yeah, a couple more came in. So Sora is asking whether it's possible to combine linear multi-epitopes to make a peptide construct which would be recognized by antibodies. That is more of an experimental question. Certainly that has been done. People have done work synthesizing what is called beads construct, where different epitopes are put together either without or with different spacers and use them to immunize and elicit responses. And this has been met with variable success. Sometimes it works very well, sometimes it doesn't. In the case of antibodies, linear peptides tend to be often a poor representation of in terms of epitopes that are conformational and discontinuous, and so and often both are the epitopes that are linked with, for example, virus neutralization and so forth. So it's more difficult to do that perhaps in recombinant antibodies, but certainly people have done that, and in many cases they have been met with success, particularly in experimental models.
Okay, and then I have the last one. It asks, are these epitopes are T-cell assay negative, which means they cannot activate specific MHC molecules or all MHC alleles? So I'm not totally sure I understand it, but I'm going to give it a shot. So if an epitope was shown to be negative in an MHC specific query, that's dependent on it being tested with the only MHC class 1 alleles being present or specified by the authors. So there's going to be a lot of data where an epitope might be negative for a whole PBMCs, and we don't know it. We have no insight about the restriction. But if they did a tetramer assay, for example, with a specific MHC molecule and it was negative, then that would be in the database. So it's limited to how the authors did the experiments. We don't, and there's a lot of information we wouldn't know. So if it was negative in the A0201 tetramer assay, we wouldn't know about B0702 if it wasn't published specifically as the allele present.
Yeah, and to emphasize what Randi is saying, exactly right. So going back to the fact that the IEDB is really an assay database, and all the data is very context dependent. So every epitope is negative until proven positive, in the sense that in reality, that the data with the IEDB has, say, a negative data curated for a particular context, so that particular epitope was negative in that particular mouse when injected as a soluble peptide. Well, that doesn't exclude that that peptide may be very positive if injected with an adjuvant in a rabbit. So it's the negativity is linked, or positivity is linked to a specific experimental context, which the user can access because it's all specified in the record associated with the negative entry. And often you have exactly that situation, that one particular peptide may be negative in a situation, but positive in another. And so that's kind of fun to see as you query the database.
Excellent. Thank you so much, Alex and Randi. I believe they are all of our questions answered for the time being. So I'd like to thank you both again for your wonderful presentations and for answering the questions. Thank you all for submitting the questions. And as a reminder, we'll have multiple of these sessions throughout the day as well. So now we're almost at time. We'll be taking a short 15-minute break. So by Pacific time standards, it's just about 3:30 p.m. and we'll be reconvening at 3:45 p.m. Pacific time, but I will pop that in the chat for you all. I'll also be sending out the share drive link. It has some resources in there. So the agenda, as I shared earlier, but it also has some flyers of information, some of our recent publications, and a link to our YouTube channel and some really interesting videos that we've made as well, if you're not familiar with our YouTube as well. So that's all in that share drive, and I definitely encourage you to take a look during the break. Alex also touched on the curation process, which is incredible, and it is highly structured, and a lot of information goes in and makes up that process as well. So obviously we couldn't go into that in too much detail for you all, but we do have a separate video that I have the link for, and I will share in the chat. I will also play this video for the last about 6 minutes of the break. So that will, I guess, be an indicator to you all that we'll be reconvening within the next 5 or 6 minutes. But with that, I would like to thank you for our first portion of the day, and we'll be taking the 15-minute break now. See you at 3:45 p.m. Pacific time. Thank you.